and thanks for tuning in to BEaTS Research Radio, a podcast and YouTube channel that aims to disseminate science and research to the community. My name is Nicole Chu, an undergraduate student at the University of Ottawa, and I'll be your host on today's episode. Joining us is Dr. Edana Cassol, Associate Professor in the Department of Health Sciences at Carleton University. She is also the co-director of the newly established Tissue Engineering and Applied Materials Hub. Dr. Cassol's research focuses on interactions between infectious diseases and immunometabolism. Her research team investigates the role of cellular metabolism in regulating macrophage function in response to infectious agents like viruses and bacteria, as well as how these processes differ across diverse tissue microenvironments. Thank you so much for joining us today, Dr. Cassol.
Thank you for inviting me. I'm excited to tell you a little more about my research.
Thank you for joining us on our channel. So to begin our conversation, do you mind sharing with us some of the main research focuses of the Cassol Lab and some of the ongoing projects currently in your laboratory?
So we actually have a sort of wide range of different projects that are happening in the lab. They all sort of center around macrophages, which are an immune cell that are part of the innate immune system that really represent one of the first lines of defense in the body. And we're really interested in understanding these cells because they're the first to recognize and respond to diverse microorganisms or infections that you might be exposed to, such as viruses or bacteria. And we're interested in understanding and investigating how cellular metabolism contributes to the fine-tuning and the regulation of these functions. So we believe that by understanding these relationships, we can find not only new ways to treat infectious diseases by making our immune systems smarter and more effective, but we also think that by manipulating these types of interactions, so the interactions between the host, the immune cells, as well as metabolism, that we can find new ways to treat non-communicable diseases such as cancers and chronic wounds. And so projects in my lab include things like using oncolytic viruses to selectively kill cancer cells, but also to reprogram the macrophages that are found in solid tumors to improve anti-tumor immunity. We're also doing work in using bacteria and bacterial products to restore the functionality through reprogramming of cellular metabolism of the immune system of aging individuals. So things like providing bacterial ligands as adjuvants to better improve vaccine responsiveness in these populations. And then we're also doing work that we're investigating sort of interactions between the wound and the bacteria that colonizes the wound and how those bacteria interact with the macrophages and drive delayed healing. And ultimately what we want to do is sort of target these interactions to restore healing processes in these individuals with chronic wounds. So
I see. Thank you so much for sharing the main research focuses of your laboratory. So at the beginning, you mentioned you work a lot investigating macrophages. For those who may not know, what are the roles of these immune cells in our body?
Right. So I'm biased. Obviously, I think that they're the most important immune cell in the body, but they're actually very interesting because they're found in almost every tissue in the body. And based on the tissue that they're found in, their functionality can be quite unique. So they play roles not only in maintaining sort of tissue homeostasis, but also they also play an important role in infectious disease and other types of diseases. So in the context of infection, in the earliest stages, they really play, as I mentioned sort of earlier, is that they're the first line of defense. So they detect the invader and they really play an important role in initiating an inflammatory response. So they send out the signals that allows for the recruitment of other cell types to that site of infection to obviously induce a more robust immune response so you can clear the infection. And so in addition to sort of sending out these chemical signals, they also play an important role in phagocytosing, so eating and killing the microbes or the infectious agents that might be in the environment to help eliminate them. And then at the end of infection, once the infection has mostly been cleared, they remove things like cellular debris or dead cells to help restore the local microenvironment. They also contribute to tissue remodeling and sort of repair processes. So they play a lot of important and diverse roles depending on the context and the disease that they've been implicated in. So.
Oh, I see. Thank you for the crash course on macrophages. So I'm sure that they're regulated in many different ways, and one of them being mitochondria. Why are mitochondria an important regulator of macrophage function?
Right. So it seems a little bit specialized. So we are interested in the mitochondria. Everyone might be familiar with them as the powerhouse of the cell. So they do contribute to regulating macrophage function through the production of energy, as well as biosynthetic precursors. So they provide all of the things that the cells need, macrophages need to become functional. So energy and proteins and amino acids to generate the effector response. But what's increasingly recognized and probably more interesting is that the mitochondria actually produces all of these bioactive metabolites and molecules that have been shown or are increasingly being shown to have potent signaling effects. So not only do they activate transcription factors and epigenetic processes, but they seem to play a really critical role in the entire functionality of the cell. So in my lab, we're mostly interested or primarily interested in the production of reactive oxygen species. So these are molecules that at low levels have been shown to sort of have direct and indirect interactions with the transcription factors or the factors that control the production of inflammatory cytokines and things like that. But at high levels, they can also cause damage to the cell, and this damage can also turn on other types of inflammatory and immune responses. So that's sort of the angle that we're investigating in my lab. So.
Oh, I see. So during these times of infectious diseases, how do viruses hijack or target the mitochondria to escape like the body's defense system or immune response?
Right. Yeah. And that's a great question. To be honest, it's very much dependent on the type of virus and the cell type that's being infected. But generally, viruses are obligate intercellular organisms. So that means they have to live within and they use all of the host cells' machinery and resources to survive and replicate. And so, really, this became initially it was really largely thought that the reprogramming of mitochondrial function by viruses was really just to support their life cycle and their propagation. But it's increasingly recognized that it may also target certain mitochondrial processes to turn off the immune response. So again, it's been shown that you can limit certain functionalities of the mitochondria to turn off things like antiviral in type 1 interferon production. And it's, as you can imagine, it's quite difficult to decipher out what the signals from the cell to turn on the immune response versus the signals from the virus to shut off the immune response. So we're sort of it's an active area of investigation. But it's actually very interesting how it's more than just reprogramming to support their life cycle.
Oh, I see. Thank you so much for that answer. And sort of switching gears to one of like another research focus in your laboratory, one of your focuses in your lab is to investigate how bacterial biofilms drive inflammation and contribute to delayed healing in patients with chronic wounds, like the diabetic foot. So how does altered cellular metabolism play a role in these processes?
Yeah, and this is, it's sort of unclear at this point, but it's clear that the reprogramming of the macrophage through the sort of remodeling of the metabolic processes plays an important role. So, I don't know how much everyone realizes how big a problem chronic wounds are. They are a significant problem. Most elderly individuals experience at some time during their life a wound that has delayed healing. And, we know a lot about what's contributing to that delayed healing. It's the types and the levels of bacteria that's found in the wound, as well as this chronic inflammation that seems to persist and can't be resolved. But the connections between the two, between interactions between the biofilm as well as the macrophages are less clearly understood. And so that's what we're trying to do. We're trying to bring together an understanding of how the types and the amount of bacteria that colonizes the wound bed and the interactions between that bacteria and the immune cells in that microenvironment prevent the healing processes. So my lab is mostly focused on the reprogramming of the mitochondria and specifically the production of reactive oxygen species. And so what emerging data really suggests, it really depends on how the levels of production of these reactive oxygen species, which turn on these chronic inflammatory processes, and some bacterial species basically reprogram the mitochondria so that they cannot be restored. So they shut them off, and it basically causes the cells to become hyperinflammatory and results eventually in cell death. So we're trying to understand how we can target these things to restore the functionality of the cells and restore healing.
Oh, I see. Thank you so much, Dr. Cassol, for giving us a greater insight on the role of mitochondria and how it's much more than just a powerhouse of the cell. Yeah, we really appreciate your presence on our channel today.
It's been
And
a
thank
pleasure to be here.
Thank you to all BEaTS Research Radio listeners for your time as well. Don't forget to like and subscribe to our YouTube, Twitter, and Instagram channels. On behalf of our entire radio team and radio director, Dr. Amelia Alarcon, we hope to see you all next week.